All right, welcome back everybody. Today I think I want to start a tutorial series on how to analyze spatial transcriptomic data. And what this video here is going to be is talking a little bit about what spatial transcriptomic data is and maybe looking at one of the data sets that I think that we could learn a little bit from in analyzing. So here we see my PowerPoint, and today I'll talk a little bit about what spatial transcriptomics is and how we can use it to answer some biological questions that we have. And it's very short, so hopefully we can get this video done pretty quickly here. So first of all, what is spatial transcriptomics? Well, spatial transcriptomics is pretty much the same thing as your regular bulk sequencing, except we now have this other component along with it. Usually in your bulk sequencing, you have your gene expression data. So it's going to be your gene counts. And usually one sample has one set of counts per gene, right? So if you have a sample of somebody's tumor, you're going to have the number of times that a transcript in your output library had mapped to, say, your p53 gene, but only one number. What we have with spatial transcriptomics is in our sample, so here on the right-hand side we can see our tumor. We have a handful of different subsamples that come from this tumor, and each one of those is going to have the characteristics that you would of bulk sequencing. You're going to have a read count number for p53 at number 1, number 2, 3, 4, 5, 6, 7, so on and so forth. In addition to your read counts, you're going to have an X and a Y location. So here on the right-hand side, we have an example of Nanostring's geomics platform, which each spot here, field of view or region of interest, is probably going to contain a lot of cells. But we do get a spatial component with this. And that component can come in things like an X and Y location, or here, relative distance to other spots. So we'll be able to calculate the distance from 1 to 2 from the X and Y location of 1 and 2. Now, there are many different types of spatial transcriptomics. They're going to range in the resolution that you get. So when we were talking about Visium there, or geomics there, you have many cells that are belonging to a single location. There's nothing wrong with that. It gives you a little bit more flexibility in where you're sampling. Next up, we would have something like Visium. So if we're going down that resolution pyramid, or up the resolution pyramid, as we get closer to the top, we get more resolution. Here on the right-hand side, we have a picture of that. And you can see each one of these little circles in the sample is going to be another sample that we're getting. So we get read counts at each one of these locations and the X and Y location at each of those. And what that does is allows us to compare the gene expression at the different locations. So we can see down here, there's low gene expression for whatever this gene happens to be. But as we go up, our gene expression is increasing. So we can do some spatial statistics with these that we'll get to in the future. Next, we have something like Cosmics and Xenium. And what these are actually subcellular transcript, spatial transcriptomics data. So with Cosmics, which is what we're going to be working with here, the data can be very big, but you get transcripts per cell, which allows you to do a lot more powerful spatial statistics. You get to do cell phenotyping, some pretty cool stuff at the cell level. And so Cosmics is the Nanostring platform, and then Xenium is the 10x platform. Each of these has a variety of genes that they can cover. The immune panel is something like what GeoMx has, or a cancer panel. If you go with Visium, you're supposed to be able to do entire transcriptomic coverage. Of course, with that, you're going to have a lot of genes that are 0 counts, but you're able to get counts for any gene on the transcriptome. But when we get to something like CosMx, we're looking at something closer to 1,000 genes, or 960, 80, if you look at the negative probes that they're including for quality control. So we get these different platforms as well as the different amount of transcriptome coverage with each of those, and you can get targeted panels for each of those. If we look at something like the difference in resolution, as we go from left to right, we're increasing the resolution. So here we have spots that contain several cells. In the middle, we get something between 5 and 10 cells, 5 and 15. But if we get all the way to that CosMx SMI that I was talking about before from Nanostring, we actually here can see the different locations within the cells that our transcripts are located at, which allows us to do some pretty cool things downstream. So we can see whether or not transcripts associated with the cell cytoplasm or nucleus are more abundant in a particular sample, or what have you. Like, there's just so much more that you can do. Transcripts co-localizing, but it's very cool. Something that many people have worked on for a very long time is how do we understand what is in our sample from the count data that we get out of our RNA-seq profiling, right? So with bulk profiling of RNA-seq, you can run through tools like CyberSort or MCP Counter, which will give you a proportion or scores of different immune cells in your sample. And that can be really helpful to associate the sample score or proportion with the phenotype or some clinical aspect of that sample. So if they received immunotherapy, do they have more T cells now in the sample from your deconvolution method? Another way to do this is spatial decon, which is something that I believe Nanostring had developed for GeoMx. You have your multiple cell types for each spot, but since you're not working with true bulk sequencing, it might be a little better to use something more tailored to it. And down here on the right-hand side, we can see the different cells and their colors. And then if we look at each one, we can see that naive B cells seem to be hot here in the left middle and the bottom middle. If we look at T cells, we can see that they kind of trail here in along on the left-hand side, as well as some up here on the top. We can see a breakdown of immune composition. There's just a lot that you can do with the deconvolution methods. Something to pay attention to here is that there is no cells being identified here on what would be potentially the background H&E image or an immunofluorescence image. So we don't see any gene expression there on our sample, right? So that makes sense. We're not going to see gene expression where potentially, depending on the technology, there are no cells, but there may be transcripts kind of falling outside of that, or just no transcripts at all, right? So here in the bottom right-hand side, or left-hand side, there's no sample at all, and we're not going to see any gene expression there, or we should not see any gene expression there. So that's more of the bulk type sequencing. But when we get to something like the cellular resolution, Nanostring has developed a tool called in situ type. And what this does is it allows you to take a single cell reference. So there's the human protein atlas, you can get single cell expression data from, and then inside that it'll have T cells, nerve cells, all that in there. And you can use that to create an expression profile. So for your T cells, what does the expression typically look like? And then using in situ type, you can deconvolve that out of your samples. So here on the right-hand side, I have one of the images from a sample of mine, and we can see that we have our cell level information, and we were able to use in situ type with the kidney cell atlas in this case, because it was for kidney cancer, and kind of deconvolve our cells, each cell, into what it is most likely to be. And we can see on the right hand side in the upper left or upper right that we have some gray cells, and that's the tumor cells in the sample. And we have this pocket of immune inflammation happening here. And that kind of matches up with what we see down here on the bottom. We have our tumor down here with the green. We have our immune inside that with the dark purple. And then over here on the left-hand side, we have far more immune, which is pretty cool. So this is one of the tools that we'll be using here because it's free, and we can download one of the cell atlases in order to deconvolve our example data. If we go now to the sample data that I think would be good for us to start working with is the human frontal cortex dataset from Nanostring, and this is going to be their CosMx SMI Spatial Molecular Imager. We can look at some of the information that they provide in the summary, and we'll be able to see all of this afterwards as well. We're going to use their summarized Seurat object. But if we look at this, we can see that they have 6,000 RNA panel containing 6078 gene targets. So this is going to be far more than what I've used in the past. So I've used their 1,000 gene panel. We can see the number of transcripts. So in our samples here, we have over 300 million transcripts detected in all of the cells, which is 194,000 cells, which is pretty cool. What's another important one? So false positives. We have some negative targets that are in our sample that allow us to see whether or not there's a high proportion of false positives being identified. False positives, yeah, which it's very, very low, which is good. Negatives target. What's another one? Scan area, we can see the total area, which is pretty big, of the sample that they had selected. So down here we can see something that we'll probably create. The UMAP projection, which will be something that we can do, as well as the transcripts located on the sample. We can do an interactive visualization if we want to go there, but keep this nice and sweet. We'll download this Seurat object. Let's go here, create a new folder, CosMx, and we'll download it here. I'll allow that download. It's going to take a while to download, but what we'll be able to do is import this into R, use the Seurat package along with some other functions and the in-situ type package to annotate our cells and be able to analyze some spatial transcriptomic data. Let's see if there's any other information on here that could be useful for us. No, I think we're good. Wonder how many samples there are, though. Doesn't say. Pre-commercial, potentially just a single sample. We'll be able to explore it when we get our data set. But anyway, so that's it for now. See you guys soon.